Recording. Hello, everybody, and welcome to our next Precisionary Instruments webinar series for our scientific speakers. And today we're honored to host Dr. or Professor Mari Shinohara, who is from the Duke University School of Medicine. She is a professor of integrative immunobiology, and she's going to tell us today about her work in evaluating cell death in the gut during acute graft versus host disease in mouse models. So a little bit about Dr. Shinohara. She's an immunologist who works on innate immune signaling in autoimmunity, neuroinflammation, and fungal infections. And she has studied Osteopontin, or OPN, for over 2 decades. And actually, it was Dr. Shinohara and her colleagues who identified that osteopontin can be actually extracellular and intracellular. She also initially identified that intracellular osteopontins function as a scaffold protein and signal transduction of multiple pattern recognition receptors. So that is a huge contribution in the field of immunology. So her group at Duke has generated a new mouse model to study osteopontin isoform-specific functions, and she is an expert in this area. So with that, I want to hand the camera over to Dr. Shinohara. Thank you for being here, and we're really excited to learn about your research on osteopontin expressions today.
Well, thank you so much, Abby. And she really gave us a perfect introduction for me, and I really appreciate it. And then I'm just going to cover pretty much lots of what Abby just mentioned. So today, obviously, I'm going to talk about GvHD. The thing is that, as Abby told us, we are interested in understanding the innate immune responses in host cells during autoimmune settings, and then also fungal infection settings. And then these days, we are particularly interested in the brain infections and then also brain inflammation. So the GvHD today, actually, this is a very first disease model we tried using Osteopontin. And then I'm going to tell you why we picked a GvHD model this time later in this talk. So let's go to the next slide. I'm sorry. Okay, so just a quick introduction about GvHD, particularly in this model I'm going to show today. This is acute GvHD disease model. So it's already known that acute GvHD can be seen in 35 to 50% of hematopoietic stem cell transplant patients. And then also it's characterized by inflammatory response, which affect the skin, gastrointestinal tract, livers, and really many other organs in the body. So the model we are going to use today is a mouse model of allograft transplant model. So first, the host is irradiated, and then we're going to transfer donor cells to host. This is a big picture. And then the thing is that this is an allograft model. So we are going to use C57B Black 6. So I'm just going to call it B6 or Black 6 mice. So we're going to take cells from those mice, Black 6, and then transfer to this bulb C recipients. And then because of the allograft model, GvHD will be developed because of the haplotype mismatch. And I'm sorry, okay, I'm having a hard time. Okay, I really have to hit it. Okay. So, but in our particular setting, we are going to use a little bit more complicated. The bottom line is the same. It's B6 cells transfer to bulb C. But here we are interested in what cells are involved. And then particularly we are interested in T cells this time. So that's why we use this particular model where we isolate CD4 T cells from Black 6 mice, and then transferred it to bulb C, particularly after irradiation. And then also another thing I just wanted to mention a little bit is we have to also transfer bone marrow cells together with T cells to bulb C, otherwise mice will not survive. And then here we depleted T cells just because we are studying T cells here. And then we are going to use wild type and then knockout T cells. So that's why we wanted to exclude T cells here. So this is only just a serving to make recipients survive. So what happens after transfer and then irradiation is like this. The first hit by irradiation really damage cells, and then damaged cells release endogenous molecule, danger molecules like DAMP or DAMPs, damage-associated molecular patterns, which stimulates antigen-presenting cells like the dendritic cells. And then also our self-antigen is presented on MHC molecule in dendritic cells, and then possibly also macrophages, and then other APCs. And then the next stage is those antigens are presented to T cells. T cells get activated, and then now T cells recognize our own antigens, self-antigens, and then attack our different organs. And then we can see the inflammation, and then also even further damages of those organs by GvHD. So this is our model. Now let's switch to Osteopontin. Just wanted to introduce our favorite molecule, Osteopontin. We have been working on this molecule for pretty many years, pretty long years. And then OPN is a protein which is a little more than 300 amino acid size, and then also known to have multiple phosphorylation sites and then glycosylation sites. So what kind of molecule further about osteopontin? So osteopontin is found as a highly expressed genes and then proteins in various kind of inflammation. That's why we are really interested, and then not only us, many people are really interested in osteopontin, because these days people run single cell RNA-seq and even not single cell bulk RNA-seq. And then if you look at different kind of diseases, infections, cancers, autoimmunity, and more, osteopontin or SPP1 gene, which is which encodes osteopontin protein, really shows up a lot. So that's why we are really interested. Other people also interested, as I said. And in immune systems, OPN is highly expressed, or sometimes not much though, but expressed in various kind of immune cells. Various kind means basically pretty much all kinds of immune cells: T cells, B cells, dendritic cells, macrophages, NK, NKT, neutrophils, you name it. And that is considered to be cytokine-like pro-inflammatory molecule. But these days we know that this is not really true. It cannot be cytokines. I'm going to talk about it. It can be intracellular, and then also can be anti-inflammatory sometimes. And then osteopontin is widely known as an extracellular protein. I would say more than 90% of osteopontin study, even 95% or even more, are about extracellular OPN. And then once osteopontin is secreted from the cells, osteopontin can be detected by receptors such as integrins, different kind of integrins, and then CD44. But there is also an intracellular isoform of OPN. So here in this talk, I'd like to call extracellular protein as sOPN for secreted OPN, and then also intracellular OPN for iOPN. So now let me just explain how alternative translation can generate iOPN. So it's at the, again, translation level, not the transcription level. So when this canonical AUG codon starts translation, what we can get is full length of OPN, which includes signal sequence, which targets the nascent protein to ER and then Golgi and then eventually secreted. So this is sOPN. And then also there is another alternative translation isoform. So starting the translation from here, the alternative site, when it showed up, its short form of OPN just happened to be pretty much at the proximal to the C terminus of a signal sequence. So this short OPN lacks signal sequence. So that's why this short OPN peptide is not targeted to ER, but instead it stays in the cytoplasm. So this is intracellular OPN or iOPN. So what do we know about OPN in GvHD? I think it's really important that OPN is now known to be highly expressed in GvHD. So that's why some papers are even suggested that OPN can be a biomarker of GvHD diagnosis and severity. However, once it gets to the mechanism, what OPN is doing, it's not known so much at all. For example, even we don't know, or we didn't know beneficial or detrimental. There are papers came out before, and then one paper says, okay, OPN is beneficial, but another paper says detrimental. So it's not really clear. The thing is that OPN can do both. Sometimes it can be good, sometimes it can be bad. So both could be really correct. It's just simply looking at different model or different side of OPN. So anyways, there are not so many mechanistic part of OPN is clear, although OPN is highly expressed in GvHD. And then what we didn't know at all is that in which cell types, because as I told you before, there are lots of immune cells, different kind of immune cells can make OPN, but which cells, OPN from which cells are impacting GvHD, either in a good way or bad way. So once it gets to that level, the detailed level, we didn't know almost anything. So that's why we thought that, okay, let's try working on GvHD. So I just want to let you know what we found here first, just to tell you the results first. So what we found is that OPN, particularly secreted OPN made by CD4 T cells are beneficial. So how does it work? Is like this. So secreted OPN from CD4 T cells are detected by CD44 and then eventually inhibits apoptosis of intestinal epithelial cells or IECs. But if we don't have osteopontin from CD4 T cells, there is really no CD44 mediated protection from apoptosis of IECs. And then those cells die, and then eventually pathogenic bacteria increases in microbiome, and then eventually we get more severe acute GvHD. So now I'm going to show you the data, and hopefully I can convince you. And then here we used compressed dome for the precision cut intestinal slices. Okay, the first experiment we did is whether OPN in T cells, CD4 T cells, are beneficial or what, or detrimental. So what we did here again, so we purified CD4 T cells from B6 mice, wild type or knockout. And then, as I told you before, we also used bone marrow cells to make the recipients survive. And in this case, we use OPN knockout in both cases, just because we really didn't want to have extra OPN hanging around in the system. The same thing, biopsy OPN knockout. So we transferred those cells to irradiated biopsy recipients. And then the data was really clear that weight loss, we found more weight loss without OPN. So this is SPP1 knockout, which is no OPN in CD4 T cells. So when we looked at that, OPN in the T cells or generated by T cells seems to be detrimental. Same thing for clinical score without OPN in knockout CD4 T cells. We also saw the more severe clinical score. Same thing for survival. Wild type behaved better in survival, but not for knockout. So we just looked at pathology pretty much everywhere, and then the most significant pathology we found in the gut. And then just a comparison here. This is just the irradiation only. And then this is for the recipient mice, so wild type CD4 T cells, and then knockout CD4 T cells. And then we found that more severe histological score when recipient mice are transferred with OPN knockout CD4 T cells, both in colon and then small intestine. And then this is just an example. In the knockout, we found more crypt loss. And then because of that, crypt regeneration was also found. And then also we saw that laminar propyria in knockout CD4 T cell transferred mice showed infiltrations of immune cells. And then we want to look at T cells a little bit more. To begin with, we just simply look at the T cell numbers and both in small intestine and then colon. And then the total CD4 T cells numbers are higher in OPN knockout, which is not great because T cells are pathogenic. So simply we found that more T cells, which is not great. And then particularly effector T cells, again, that's even worse. We found more T cell infiltration and the recipients transferred with, again, knockout CD4 T cells. But as you know, T cells have different kind of T cells, particularly T helper cells, pathogenic Th1, Th17, and then also regulatory or more anti-inflammatory Tregs. However, those numbers really didn't change. So it's not really the quality of T cells, OPN changed, but the recruitment seems to be different here. And then next we looked at the cell list. So here is just a simple slice culture of small intestine and the colon. And then we stained with DAPI and the tonal. And then if you see this green signal, that's the tonal positive cells, which means those are cells which are dying. And then we just found more tonal positive cells in the recipients transferred with knockout CD4 T cells. The same thing with colon. So this data suggests that OPN in CD4 T transfer CD4 T cells limits cell death in the gut. The next thing we tried is looking at the microbiome. So we looked at microbiome composition by sequencing 16S ribosome gene from day 7 fecal samples. So we had 5 different groups. So the first group and the second group are simply the naive wild type and then SPP1 recipient mice. We just wanted to know whether simply wild type knockout, total knockout, they have any difference without any disease. And it's blue and this yellow lines, but we really don't see so much of difference here. It's pretty similar. And then we irradiated SPP1 knockout mice. This is the recipients we use in this model. And it shows a little bit of changes, but still it's not really widely shifted from here. But once we induce the GvHD, both in wild type or knockout transferred CD4 T cell recipients, we found pretty big shifts from original distribution of this microbiome, components of microbiome. And then also we can see the difference separated phenotype between wild type knockout CD4 T cell transferred mice. And then particularly here, if you can now just focus on those 2 GvHD mice, we found the different types of bacterial odors, particularly, for example, like these purple ones, enterobacteriales, are really induced after GvHD induction. And here, I just wanted to let you know that at this point, we don't know. We still don't know whether this is just simply the results of inflammation induced by GvHD or this changes of microbiome is actually feeding into or somehow limiting the inflammation. We still don't know, and probably it's more like egg or chicken or egg kind of argument here, but just wanting to show this is what we got. And then here at the genus level, Escherichia was increased in GvHD mice compared with naive and irradiated mice. And this observation correlates with previous studies where the GvHD microbiome is characterized by an overabundance of the Escherichia genus. However, OPN did not appear to regulate these features of GvHD-associated microbiome here. It's pretty much similar here, but once we looked at the different kinds of bacteria, we could see the difference. For example, here, OPN greatly enhanced the levels of health-associated commensal bacteria, Akkermansia. The lower levels or loss of Akkermansia have been associated with an increased risk of development of acute GvHD. So this really goes very well with previous finding, which showed that severe GvHD correlates to lower levels of Akkermansia. And it is also known that Akkermansia protects intestinal barrier. So it's a good bacteria here. And in contrast, OPN from CD4 T cell significantly reduced the overall level of commensal bacteria of bacteroideals. Right here, it's reduced. And then the reduction with OPN is reflected in the trend of major genera of bacteroideals right here. And then those bacteria are known to be pathogenic in GvHD setting. So it's really correlating to what people found before. And then here's a little bit of details. So as you can see here, so this is a bacteroidetes genera. So those are also increased in host small intestine in CD4 T cell knockout transferred recipients. And then here is a host gene expression. So you can see the difference in DFA5. This is differentiating alpha-5, which has been previously known to go together with bacteroidetes. It seems like the epithelial cells regulates the abundance of bacteroidetes through expression of DFA5. So now this probably means that because in the knockout T cell transferred mice are really struggling with very severe acute GvHD. So that's why host cells are responding to really control over it. So that's why the DFA5 expression is higher. So that's what we found. The bottom line here is that the microbiome got really also impacted simply by, I don't know whether I can say simply, but just the OPN only expressed by T cells, particularly CD4 T cells. So impact of osteopontin only from CD4 T cells is really a lot. So here, just going to the next next part of the story, I just want to go through very quickly, and I'm not going to tell all the details because this is such a busy slide. What I wanted to let you know here is, here are iOPN and then also sOPN, just because the localizations of those two isoforms of OPN are different, their functions are completely different. And particularly iOPN is downstream, involved in the downstream signaling pathway in different kind of pattern recognition receptors right here. But sOPN, they are secreted. So of course, they are not going to be in the cytoplasmic signaling pathway. But instead, sOPN is already known to be detected by some integrins and then also CD44. So please just remember this part because I'm going to get back to that later. Now, So how can we tell the difference, iOPN or sOPN's involvement in some disease models in vivo? So to understand that, we developed this system. So please just look at D and D first. So the bottom line here is that we developed two different kinds of new mouse lines. So one mouse line only express iOPN under Cree expression. So here is intracellular OPN shown with red. And then this E is the mouse line, which express only secreted OPN in a certain Cree expressing cells. And then we also have wild type. In wild type, of course, it's global. So the cells which can express OPN can just express it. And then knockout is total knockout. And then, so this is a control for D and E. Without Cree, there is really no expression of OPN. And I'm going to explain a little bit about this system. So here is iOPN and then sOPN generation. With or without signal sequence, just because of alternative translation start position. And then how we made iOPN only or sOPN only expressing system. So if you can look at the first one, so this is endogenous OPN. As I told you, there is an OPN signal sequence here, and then around in terminus, there is an alternative translation start site, also it's shown here. To generate iOPN only, we made a mutant which completely lacking the OPN signal sequence. And then anything we can get from here just stays in the cytoplasm. And for sOPN, it was a little bit trickier, but it's not so complicated. What we did is we simply replaced OPN signal sequence to IL-2 signal sequence. Why IL-2? There are two reasons. It doesn't need to be IL-2 though, but the reason we picked IL-2 is one, IL-2 is just a signal sequence, it's just really good. And then it's just ensuring to get into the ER. And then two, reason two is more important. IL-2 signal sequence really do not start alternative translation start site here. So anything starts from here, not from here, and everything from here turns out to be all sOPN molecules. And then a little bit about Cree locus system. And here we particularly use LSL or lock-stop-lock Cree system. So this system is not really depleting the genes, but instead depleting the stop sequences here. So what happens is that without Cree, stop signal is here. So that's why we really don't have any SPP1 or OPN production or transcription, no transcription. But once we cross the mice with Cree expressing, genome, the Cree protein cleaves locks here. And then stop gene is gone. And then now with Cree, we can get the transcription and translation of this part of the gene. And then eventually, as I told you before, if we pick the iOPN system, we can generate the iOPN only, and then also sOPN only by using the other model mouse line. So here is a gene, wild type, OPN, endogenous sequence, iOPN, we completely got rid of it. And then sOPN, this green part is IL-2 signal sequence. So using this model, we wanted to do the experiment, but validation is important. So we first tried iOPN expression validation. So three different groups. The first one is wild type. Second one is OPN knockout, total knockout, no iOPN, no sOPN. The third one is a new mouse line we generated, LSL iOPN, expressing only iOPN under control of BAV1. So BAV1 Cree is active only in hematopoietic cells, which means basically most of the immune cells, including T cells. So here, wild type serum OPN from the mice. Yes, we can detect OPN, but OPN knockout, of course, there is no OPN. And then number 3, which only express iOPN. So that's why we didn't see OPN in serum, which is good. And then the next one, similarly, we simply plated bone marrow derived macrophages, and then just looking at the supernatant, basically looking at whether sOPN is there. Number 1, yes, sOPN is there because wild type makes sOPN, iOPN both. But number 2, no iOPN, sOPN. So of course we didn't detect sOPN. And then number 3, it was good. We didn't detect sOPN. And then importantly, in a cytoplasm, so we separated cytoplasm from the cells. And then we found, in this case, we are detecting iOPN. And then number 1 and number 3, wild type, and then LSL iOPN, CRE, we detected OPN, which is in the cytoplasm, so which is working well. And then also you can see that the model system here, the expression level is pretty similar. It's not like overexpression just because we preserved the OPN promoter sequence. So again, really no overexpression. And then also the validation of sOPN, and then also validation, no iOPN in this system. Again, similarly, OPN, and then I'm sorry that we just flipped the order. The pink color shows sOPN only. And then also LSL sOPN. This serves as knockout. Supernatant, again, this most important one, sOPN only. Yes, we can see OPN. Importantly, cytosol, we didn't see cytosolic OPN in this sOPN only mutant. So it's working well. We also tried microscopic analysis looking at sOPN. It's really targeted to Golgi here. And then this is with CRE LSL sOPN. And then here we also use LSL sOPN without CRE. In this case, it serves as OPN knockout. So we have a little bit of background, but basically no targeting, no yellow color here. Yellow color here. So now we use those models and then run the experiment. First, we tried sOPN mice, and then we found the difference here. So the red line is LSL sOPN. Again, this is serving as OPN knockout. And then the black line shows the mice expressing sOPN only. But again here, we used only T cells from those mice. Otherwise, background is all OPN knockout. So again, the source of sOPN alone in this black line mice are the recipient, I would say, are only from CD4 T cells. And then we found the difference here, and then even survival. So this really tells that CD4 T cell, OPN generated by CD4 T cells, are protective. And then here also we wanted to try iOPN in CD4 T cells, but we didn't see the difference. So intracellular OPN in CD4 T cells is not really involved in here. So now we try to focus on sOPN from CD4 T cells in this model. And then I mentioned before, sOPN can be detected by integrins and then CD44s. So the next we asked which receptor is involved. And here we try both, and then it turned out to be CD44 is involved for the protection. So here the compressome comes up. What we did here is we have the biopsy knockout mice OPN. Here, it's not irradiated and not T cell transfer, just a healthy naive mice. Took out the gut, small intestine, and then using the compressome, we generated a 400 micron thickness of samples and then plated in tissue culture. And then here in this tissue culture, we treated those tissues with recombinant TNF alpha to induce apoptosis. It's just known very well that TNF alpha is a really good inducer of cell death. So we tried that. And then we wanted to rescue with recombinant OPN. Again, recombinant OPN is added from outside. So that's secreted, not intracellular. So this means sOPN as well. And then also we use CD44 antibody to block CD44 receptor to prevent OPN to be detected by CD44. And then here is the results. So here, toenail cell numbers are like 300, but when we treat tissues with TNF only, we saw more cell death, toenail positive, and we added sOPN as a recombinant OPN, and we could really limit the cell death here. But when we block CD44 by antagonistic antibody, then we could rescue the cell death, induce the cell death phenotype of TNF here. So this data told us that secreted OPN from T cells are detected by CD44 to limit the cell death. So now we want to look at how the sections, and this is how it looks. Untreated recombinant TNF treatment only, recombinant and OPN, and then also blockade. So as you can see, toenail positive cells are shown as red or pink. And then as you can see, lots of cell is going on in IECs, but adding recombinant OPN or sOPN reduced toenail positive cells. And then now we blocked CD44 receptor. And then now we started to see the toenail positive dying cells again. So now the summary right here. So OPN generated by allogenic CD4 T cells limit acute GvHD is manifesting in the gut. And then a mouse system for conditional OPN isoform-specific production is developed. And then importantly, sOPN, not iOPN, protects IECs from cell death. And then we also found that CD44 mediates sOPN's protective role in acute GvHD. So just the acknowledgement, Nupo, previous postdoc, and then also Elizabeth, previous MSTP students did this work. Nupo is really the spearhead. And Elizabeth was really helping T cell transfer and also the microbiome data analysis. And then these are the people in my lab. And then also I really want to appreciate collaborators at the University of Iowa, Dr. Mangalam and then Dr. Shahi. So thank you so much. And that's all.
Thank you so much, Dr. Shinohara. I want to open up the floor now. A couple of our colleagues are also on the webinar, and see if anybody had any questions. Please feel free to unmute yourself and ask questions. And don't worry, if you're a little shy and don't have questions today, you can always send a message to me and I can send it over to Dr. Shinohara and we can put you in touch. Wonderful. So Dr. Shinohara, we don't have any questions right now, but hopefully we'll be able to follow up. Thank you so much for your time and for your work and for sharing it with us. If you need anything, please reach out to us at Precisionary as well. Thank you.
Thank you so much. Great to see you.
Bye-bye. I will be in touch. Thank you.
Thank you.